আমি ইন্ট্রোডিউস করি। গুড ইভিনিং এভরিওয়ান, সবাইকে আজকে স্বাগত। আমি সৌম্য সরকার আজকে আপনাদের সাথে। আজকে সবচেয়ে বড় কথা, আমাদের সাথে প্রেজেন্ট আছেন ডক্টর শুভাশিস ঘোষ, সেই সুদূর আমেরিকা থেকে। উনি হেড অ্যান্ড নেক ক্যান্সার ল্যাবরেটরিতে আছেন হাওয়ার্ড মেডিকেল স্কুলে। এবং আজকে আমাদের অনুষ্ঠানটা বেসিক্যালি সায়েন্স পপুলারাইজেশন, এই ব্যাপারটাকে, সায়েন্সকে বাঙালিদের মধ্যে পপুলারাইজ করার জন্য আমরা এই ইনিশিয়েটিভটা নিয়েছি। এবং সেখানে ডক্টর শুভাশিস ঘোষ যে সাহায্যের হাতটা বাড়িয়ে দিয়েছেন, আমাদের সাথে আজকে যে এই মঞ্চে উপস্থিত থেকেছেন, তার জন্য সত্যি আমরা কৃতার্থ। এবং আজকে আমাদের এখানে যারা ছাত্রছাত্রী আছে বা যারা দর্শক আছে, তারা ডক্টর শুভাশিস ঘোষকে আজকে কাছে পেয়ে আশা করি খুবই আনন্দিত হবে। এবং আমরা এই অনুষ্ঠানের প্রথম ভাগে আমরা উনার যে সায়েন্টিফিক যে কাজকর্ম, মানে উনার পিএইচডি বা উনার যে রিসার্চ কেরিয়ার, বর্তমানে উনি যে রিসার্চ করছেন, সেই রিসার্চ সম্বন্ধিত অনেক কিছু জানবো, কথা বলবো। এবং পরবর্তী ভাগে, দ্বিতীয় ভাগে আমরা উনার যে এই জীবনের যে জার্নি, হ্যাঁ, ফ্রম স্কুল টু কলেজ, কলেজ টু ইউনিভার্সিটি, ইউনিভার্সিটি টু পিএইচডি, পিএইচডি থেকে পোস্টডক, এই পুরো জিনিসটা কিভাবে কোথা থেকে শুরু হলো, কোথা দিয়ে গেল, কিভাবে রিসার্চের মোটিভেশন এলো, ক্যান্সার রিসার্চ কেন, এই জিনিসগুলো আমরা আস্তে আস্তে সব জানবো। শুভাশিস, তোমাকে হ্যান্ড ওভার করছি।
প্রথমে অনেক ধন্যবাদ। থ্যাংক ইউ for giving me this opportunity to come here and present my work and share my journey. So it is like wonderful for me, and as well as I'll be happy to answer any kind of queries or questions regarding my journey and overall work. So thank you. A big thank you for giving me this platform to share my work. So shall we go now to...
Yeah, we'll start the presentation.
Okay. Okay. Thank you. So I have done my PhD at National Institute for Biomedical Genomics, that is called NIBMG in a short form. And my title of the PhD work, just for like you to know, so this is like deciphering the significance of notch pathway related mechanisms in gingivocal oral squamous cell carcinoma. I have done my PhD under supervision of Dr. Sandeep Singh. He is a renowned scientist and doing great work in oral cancer biology in India. And I was registered with University of Calcutta for my PhD registration. As a journey, you can see just a like informal slide I am taking. Like I have done my schooling from Iron Hemangini Vidyayatan High School as well as Raghunathganj High Schools. Both are in Murshidabad. And then I have graduated from Jangipur College under University of Kalyani and doing honors in zoology. Then I came to University of Kalyani main campus at Kalyani doing my master's degree in biotechnology. And after my MSc, I have qualified 𝑑BT-JRF exam for my pursuing my PhD degree. And for that, I have enrolled with National Institute of Biomedical Genomics at Kalyani. So after completing my PhD, I recently submitted my thesis last year, December, and then I applied for postdoctoral fellowships. And recently, I got into Massachusetts Eye Infirmary, that is under Harvard Medical School, for my postdoctoral fellowship. So right now, I am doing my postdoctoral fellowship in Mass Eye at Harvard Medical School. Coming back to my research topic, so what is cancer and how it works? So as you may have heard, that cancer is a disease of uncontrolled proliferations, where cells are basically proliferate uncontrollably, where the mechanism of breaking the cell cycle or stopping the cell cycles are not functioning, and cells can grow and form tumors. So cancer and tumors are the terms you may have heard already. So these are the most common terms generally we use for cancer. And nowadays this disease is very prevalent, basically in India. So we all have heard about cancer, like in our families or in friend circle, so or in our village, town, everywhere. So I have worked in this particular disease. So talking about cancer, so recently there are two renowned scientists, Robert Weinberg and Douglas Hanahan. So they tried to basically summarize the basic or core characteristics of cancers, and throughout the years they have uh, they have observed that there is a adding new features or characteristics of cancer, and they then tried to summarize the overall characteristics of cancer cells in a hallmark of manner. So you can see in these charts, so there are total kind of 14 basic characteristics. Through those they have tried to define cancer as such. So talking about that, you can see that these are the features. Like cancer cells will evade the growth suppressor. Growth suppressor means the cancer cells are able to breaking or skip the normal suppressing mechanism, and then they will grow uncontrollably. There will be genetic changes. So overall there will be, as you have heard, DNA. So there will be mutations at DNA, at genome level. There will be resisting cell death. They will through normal mechanisms, cells will eventually die after certain amount of time, but in cancer cells will not die. So basically they are resisting the cell death. As well as there will be deregulation in cellular metabolism. Cells, cancer cells, as they are growing faster, so they need more kind of nutrients and overall food, you can say. So their alteration in metabolism is a one of the characteristics. As well as they unlock phenotype plasticity. That means that they are very dynamic in nature, they can change their state, so it is very difficult to target them with the already available drugs. As well as there will be so as we know, there is immune system in our body that basically targets whatever the external or outer particle or any kind of agents enters in our body. But in case of cancer, the cancer cells are so smart that they can avoid our immune system response. As well as they have all gain other characteristics that includes the tumor promoting inflammation will be there. So in that sense they are, again, they kind of grow and adjust with that overall microenvironment. And there will be microbiomes that also helps cancers to grow. They can modify the overall blood vessel formations, as well as they can invade and metastasize to other normal healthy organs. So these are the basic characteristics of cancers. So it known as hallmarks of cancer. So you can go through this review. It is very fascinating, and to understand the cancers as a whole, or as a understand the biology of cancer. So it is kind of a very popular and good review article. Going further, so I have worked in oral squamous cell carcinoma. So as a starting and how this cancer progress, I have taken these slides from MPI. So that means that oral cancer is a cancer of mouth, basically. So there is epithelial layer in our inner mouth, basically from inner gums, cheeks, and from the tongue portion. So how it starts? So basically there is the epithelial squamous layer. These cells you can see. So cells are like this. Then there is alteration. Alter cells means there will be some alteration, specifically like genetic events that can happen, like point mutations, deletion, copy number alteration. These are the genetic events or alteration of genomes. So from that hits, these cells got some specific characteristics. So then throughout the process there will be steps like precancerous stage, as well as at the final stage there will be invasive cancer. Invasive means the cells can be there, as well as they can try to invade other organs, or they can metastasize, they can spread to other part of the body. So apart from genetic events, in the whole process of tumor progression, basically how the tumor goes to from a benign to a more malignant state or spreading to other parts. There are events like epigenetic events that include DNA methylation, histone modification, mRNA modification. These are the steps, basically events that happens. And as a risk factor, you can say that overall lifestyle is a ma- lifestyle that matters, as well as consumption of alcohol, tobacco, HPV infection, and pollution. These are the risk factors. So it is not like that someone, if someone having smoking or having tobacco, he will eventually develop cancer, but the chance or the risk is maximized in those people. So in that sense, these are the risk factors. So as we know that cancer is a factor disease, multifactorial disease or complex disease. So that means there are many factors that ultimately gives the origin of a particular tumor or cancer in our body. So I have done my PhD in oral squamous cell carcinoma, as I have already told. So why this particular cancer? Because in India, in case of specifically in case of male, this is the most prevalent cancer, basically the cancer of lip and oral cavity. And at the same time, it is the second most cancer among Indian population in case of both male and female. This data is from India Globocon data. That is the organization that basically year by year they update the overall cancer prevalence and frequency, those stuff of data worldwide. So I have taken this data from Globocon India 2020, and that shows that oral squamous cell carcinoma is kind of pandemic, you can say, for Indian population, and it is like increasing. So I thought this is very interesting topic to work on. So as I've taken this, am showing these figures to show that the habit of tobacco chewing, as we all know, that this is a very common or popular things we Indians are having throughout the years. So this tobacco is basically act as a risk factor, as I have already told, and it works in a manner that it inflame our overall oral cavity or a mouth. And as we are keeping this tobacco in our between our teeth and gums, that inflame the overall that particular part of our body. And throughout the years, these tumors basically through in some patient in some people that originate in that particular region of our mouth. So this is a very common cancer in case of Indian population. Talking about its aggressive nature, so studies done by Tata Medical Hospital, Mumbai, they have studied patient around 2,275 gingival oral cancer patient, and there they are showing that the recurrence rate means after certain amount of treatment, certain time of treatment, the cancer again come back. Eventually it becomes more aggressive, and the follow-up studies are showing that the patients in which the cancer comes back, they will only survive 2.7 months. So this is a very, very, very aggressive cancer, as you can see. So to study this cancer further, we took the approach of personalized medicine, or you can say precision medicine or specific medicine. What does that mean? That means that we already know that there are many kind of cancer drugs throughout the world. Scientists are working, researchers are working to find out the drugs to treat this particular type of cancers. But as you know, and you may have heard that in our vicinity or in our surroundings or in our neighborhood, that though patients or uh, the patients initially are responding to a particular drugs or treatment, but eventually the cancers comes back. So that's why this disease particular so tough to treat. So from that perspective, throughout the global research ecosystem, people are trying to find out the root cause of this resistance, and why the drugs are basically not effective to kill those cancer cells in a particular patients. So it has been found that we all are unique. Basically, we all have a particular particular structure or the set of genes that works in a unique way. So in that sense, the scientists are trying to see that why in some patients these particular drugs are responding, but in others they are not. So for that purpose, they are trying to see that at the molecular level means at the level of DNA, RNA, and proteins. So you can see there are primary tumors. Sorry. From the primary tumors, eventually, as I already told, the cancer may spread to other parts. They become metastatic. So that process is called metastasis. So from the benign tumor, that is primary tumor, eventually cancer cells move to other places or healthy places of the body or organs, other healthy organs, and then again they form the tumor in that healthy organ. So that process is called metastasis. So scientists are doing researcher what they're doing, they are trying to take the tissue or cells from both primary state and at the metastatic state, and then they are doing this particular, these are the methods basically for precision medicine. They are doing paraffin tumors. So paraffin tumors will eventually give us the result that at the protein level or at the cellular level what are the changes are happening by doing immunohistochemistry. These are the common methods, and at a large scale, these methods are being used by researchers throughout the globe, as well as liquid biopsy, the collecting the blood samples from patient, as well as patient-derived organoids. These are the kind of mini tumors growing in a lab culture conditions. I have particularly worked in this type of system. I will eventually show you. As well as patient-derived xenografts means the tumor samples from patient can be injected into a mice model to see its progression and how it overall form a tumor in a mice model to see its aggressive nature and all that. At the same time, they are doing basically applying NGS, as you may have heard, next generation sequencing. By that, basically DNA and RNA sequencing are done, as well as at the proteomics level also. At the protein level also, people are doing NGS methods by doing mass spectroscopy and those methods. So all of these methods are basically telling us that at the molecular level, at the DNA, RNA, or protein level, how each patients are unique to each other, and it will eventually tell us what are the molecular target we can see, and at the patient level we can target it differently so that we can have a better result for each patient. So this is the concept basically. So from this point of view, I have taken my problem. So for oral cancer also, as I have worked in NIBMG, and NIBMG is kind of part of international project that basically sequence cancers and overall trying to find out the molecular signatures. So that part is done by India project team of ICGC. ICGC means International Cancer Genome Consortium, and at NIBMG there is a particular group, they are doing the sequencing part. So they have in 2013 they have published a paper basically taking the DNA samples from Indian patients and doing at the local level, and they have find out that there are few driver genes means the driver mutation means the mutations that basically drives a tumor. So that means for forming a tumor and its progression, that particular mutations is playing some important role. So in case of oral cancer also, we have find out there are few of the genes that are basically mutated, and in those list I got hit like have seen that there is a particular gene called notch1. Notch1 genes got significantly mutated. So as you can see through this arrow I have shown that there are so this green, red, and blue are these three particular group of oral cancer patients. So and in those particular group, these are the main genes that are got mutated, that is caspase-8 phaton in a particular group, TP53 or P53, the guardian of the genome, as you may have heard. So that got mutated, and the other group includes other some of the other important driver genes mutation. So throughout these three group, there is a common genes, and that is Notch1. So, and interestingly, that Notch1 mutation in case of head and neck squamous cell carcinoma, like oral cancer, in which I have worked, is completely different. The mutations are basically located to the extracellular domain of this particular receptor molecule. So these are receptors basically, they are in cell surface, and these mutations are basically inhibiting the ligands to bind and basically inactivating this particular notch pathway. But in case of other cancer like leukemia, that is blood cancer, and breast cancer, the mutations are basically localized in the intercellular domain. So in that sense, the mutations in case of oral cancer, try to find out that it is very different. So as a functional consequence, how these mutations are important and how it is giving the aggressive nature of oral cancer in Indian patient. So those are the basic questions we asked initially in my PhD journey. So to coming to the cancer stem cell model. So to address those questions, I have adapted cancer stem cell model. Cancer stem cells are a very rare population of cancer cells. Those have the ability to self-renew itself, as well as they can differentiate and form the overall tumor. And it has been find out that cancer stem cells are kind of the root cause of relapse or recurrence or coming back of tumors. How they do? Like, as these cells have the characteristics of slow growing. So basically, when we target cancers, as we know, the cancers are kind of fast growing cells. So if we target cancer, the fast growing cells will eventually die because they are growing fast. They will take the drugs or treatment more robustly. So they will die. But the cancer stem cells, at the response of drugs, they will slow down their proliferation. They will proliferate slowly. So eventually they will take drugs more less. So they will kind of survive. So this is their survival strategy. And then to correlate cancer stem cell with Notch pathway. Notch pathway is a signaling pathway basically that works in a juxtacrine mode. That means two adjacent in between two adjacent cells, there will be certain kind of ligands, as you see, JAK1, JAK2, DLL 1, 3, 4. These are the Notch pathway ligands. And receptors wise, Notch 1, 2, 3, 4. In case of mammals and humans, basically, there are four Notch receptor, Notch 1, 2, 3, 4. So whenever there is signaling between two adjacent cells, the adjacent cell will work as a signal sending cells, and the cells which act as a signal receiving cells, in which these are the receptors, and eventually that signal works, and in downstream they will activate some downstream genes. This signaling is prominently working in developmental biology as well as maintaining stem cells in normal scenario. So from that perspective, as I got mutation in these genes, and cancer stem cells, in cancer stem cells, Notch pathway is important. So I tried to find out the relationship between Notch signaling in cancer stem cells. So these were my basic observation. So that are inactivating mutation in Notch genes are present in oral cancer, and at the same time, Notch pathway is conserved stem cell regulator pathway in multiple tissues and in developmental biologies. So from this observation and questions, I have hypothesized my PhD project that was inactivation of Notch pathway may facilitate a more aggressive nature in oral squamous cell carcinoma by modulating the stemness. So based on the hypothesis, these are the objectives which I have to work on. So to assess the level of Notch pathway related genes in primary oral tumor derived cancer cells, to study the functional consequences of altered Notch genes related to different cellular functions, to examine the possible mechanism of reciprocal interactions between diverse cell types in controlling Notch pathway related to signaling in oral cancers. So for my study, I have employed spheroid culture model. What is spheroid culture? Basically, spheroid culture. So in lab to work on in cancer and like other disease, we usually culture cells in plates. Basically, By giving them medium as their food, they will take the nutrient from the media, and we at 37 degrees Celsius the body temperature kind of to maintain the cells. So in those conditions, so oral cancer cells are basically epithelial cells, but to grow the stem cells from that particular population, we employed this sphere culture, where basically we are trying to see the stem cell growth. So we plated cells in a single cell mode and in a suspension mode. So basically they will not attach to the surface of the plate, rather they will be in a suspense medium, and they will eventually form the tumor. So you can see these are the growth from a particular single cells. So you can consider these cells, these particular spheroid, as a mini tumors growing in a plate culture condition. So to see the downstream things, that if we target these things, and if you inhibiting our particular targeting pathway, then what is happening. So for that purpose I have taken this taken help from for this particular culture method. And we have also seen that in this particular culture method, compared to the adherent, the conventional culture which people usually do, in this culture method there is enrichment of CD44. CD44 is a known stemness marker for stem cells in oral cancer. So eventually found out that there is CD44 high. This violet graph portion is showing CD44 high expression compared to the black portion. Black portion is for adherent conventional culture, but in spheroid culture there is a upregulation for CD44 gene. At the same time, we tried to find out that in sphere culture, what are the alteration or upregulation of the known stemness regulator genes, that is LDH1A1, SOX2, KLF4. These are the known stemness regulator factors. You have heard in many articles and research publication about this, but known factors. And at the same time, throughout these three different OSCC cell lines, there is upregulation for Notch1 gene also. So that means in sphere culture, as stemness regulators are getting upregulated, at the same time, Notch pathway genes are also getting upregulated. That means that Notch pathway is important in maintaining stemness of oral cancer cells. Then, as there was mutation in patient, like there is inactivating mutation, so to mimic or to recapitulate that process in our in vitro culture model, we have taken help of this LY and RO. These are basically Notch pathway inhibitors. They target the gamma secretase inhibitor enzyme that is very important in maintaining or modulating the downstream Notch signaling. So these particular small molecules can target those target gamma secretase, and in that sense, they will inactivate Notch pathway. So in OSCC cell line, that is SCC070, that is a OSCC cell line, where I have shown that these particular inhibitors can inhibit Notch pathway. So you can see compared to this bar, this is the control graph bar, and compared to the control bar, there is significant reduction. Reduction is there is suppression of this particular Notch genes, Notch1, H1, H1. These are the Notch1 is the receptor, and H1 and H1 are the downstream, major downstream gene of Notch pathway. So that means these small molecule inhibitors are inhibiting Notch pathway. And the same time, we have taken help siRNA mediated small interfering RNA. That means, and it basically transiently knock down genes at mRNA level. So that is also showing it efficiency when we target siRNA to target Notch1 as well as HES1. So those are showing significant down regulation of Notch pathway. So that means this in vitro methods of employing small molecule or siRNA is like effective to downregulate the pathway. So we can mimic the patient level Notch inactivation in our culture medium, in our culture condition basically. So to see that if we target Notch pathway, what happens to our this mini tumors or spheroid, you can see. So if we are targeting throughout these three different OSCC cell line, their oral cancer cell line, so there we employed this LY and RO. These are the small molecule inhibitors for gamma secretase or Notch pathway at 5 micromolar level. So you can see there is no reduction in sphere formation. Compared to control, there is even high sphere formation. See, so that means that at the spheroid level, if we target Notch pathway or inhibiting Notch pathway, then there is no reduction in sphere formation. Rather, the cells can retain their stemness and they can grow as a spheroid. And the similar kind of result has been also observed in case of siRNA mediated inhibition, where we target Si notch, notch by siRNA, and H1 by siRNA. There is also significant kind of sphere formation compared to the control. So that means notch inhibition leads to a higher frequency of sphere formation of OSCC cell lines. So rather than the remission or suppression of sphere formation, notch inactivation is leading to a more aggressive. They can proliferate more and they can form bigger spheres, as you can see in this particular condition. So to understand the biology of Notch pathway and its inactivation, how the tumor is behaving, we also tried to generate, we basically generated clones. Clones means, stable clones means their Notch pathway will be constitutively active or like continuously active or continuously inactive. So those conditions we have generated in our lab to see the biology of this particular pathway. So this GFP or green color you are seeing, these are the cells sorted after generating these stable clones. Stable clone has been generated by lentivirus mediated transduction process, where hex cell mediated lentivirus has been generated, and then again they are injected into uh, OSCC cell lines to generate this particular clone. Clone are basically genetic modification of a original cells, and by that means we are trying to kind of overexpress or downexpress a particular gene or protein. So going further, as the small molecule results are showing that inhibiting Notch pathway is not altering at all sphere formation, rather they are higher sphere formation. The similar kind of result has been observed. So P-lenti is the control condition for both the cell line. HICN and HS is the wild type Notch pathway active state, you can say, and BH is the mutated H for the Notch pathway. So this condition, the red condition, is the mutated one. So you can see both for sphere number and sphere diameter. Both these parameters, you can see in the red condition there is higher number of or bigger number of sphere formation compared to the other conditions. So that means that inhibiting Notch pathway is not basically taking the characteristics of sphere formation. Rather, the cells can be more aggressive and they can form more aggressive spheres. So to find the results in vivo condition means that if when we try to see our result in kind of in a living system, like it could be mice, it could be zebrafish, it could be rabbit. So for that purpose, we have taken help from the zebrafish embryo in our zebrafish facility at NIPMG, where we injected this GFP, this green, basically the green patches you are seeing. These are the basically tumors. So we injected we first modified the cells based on Notch pathway over continuous high expression and continuous low expression, and then we injected. So the BH is continuous notch low condition. So there you can see there is the green patch is kind of bigger, or it is taking larger portion of their body. So that means targeting notch pathway is not basically helping in this condition, and rather it is giving more aggressive tumor formation in zebrafish model also. And at the same time, you can see this line graph is also showing the red line is basically continuous notch inactive condition. There is the probability of survival of this zebrafish embryo after injecting these particular cells is very less compared to the other conditions. So that means the zebrafish embryos are also dying due to the tumor of notch inactive conditions. Then to see the role of Notch pathway in drug resistance, we have employed cisplatin drug. Cisplatin is a major platinum-based compound or drug basically for treating oral cancer patients. So there we are seeing that if we target or treat these primary spheres or organoid from oral squamous cell carcinoma cell line, oral cancer cell line, so then this there is no reduction in viability. And this 2' means the second generation. So basically we targeted the spheres generated from oral cancer cells, and then again we treated with cisplatin, and then again collected and again plated for again next culture. But throughout this culture, cisplatin treatment is not killing those stem cells in spheroid culture, rather they can maintain their state. So then we try to find out the reason or cause behind this resistance. So for that purpose, we have taken spheroid culture from both primary and cisplatin treatment and secondary cisplatin treatment, and then we try to find out the gene expression at the mRNA level. So these are the primary sphere, these are the secondary spheres. In both the condition, I kept control and cisplatin treatment, and as you can see that there is a significant upregulation due to cisplatin treatment for Notch pathway genes, Notch1 and H1, CMYC and SOX9. These are the SOX2. These are the already known stemness regulator. Those are significantly upregulated, as well as CK genes are cytokeratin genes. There is kind of variation of expression, but mostly the cytokeratin which are related to basal or stem cells are getting upregulated. So that means due to cisplatin treatment, there is significant upregulation for Notch pathway, and in that sense, Notch pathway basically conferring the resistance against cisplatin, it acting as a major factor. So to see the involvement of Notch pathway in cisplatin resistance, we took help of the clones that we have already generated for these two particular cell line, oral cancer cell line, and the BHAS is the continuous Notch inactive clone where there is significant reduction of IC50 value for both these conditions. So IC50 value is that value where 50% of the population or cell is basically dying. That is kind of very known parameter in pharmacology or drug screening or compound screening. So we have measured that thing. So we have found out that if we are continuously inactivating Notch pathway in oral cancer cells, then there is significant reduction of this particular IC50 value. That means cells are becoming more vulnerable if we target the Notch pathway along with the cisplatin. So that is the overall theme, that due to cisplatin treatment, there is upregulation for Notch pathway, and if we target Notch pathway, then there is significant reduction. So to see it further, then again we have taken help of these Notch pathway inhibitors, LO and RO, that I have already told. So this is the control condition of primary sphere, where in cisplatin, as you can see, there is no such alteration due to cisplatin treatment. LOI only is not diminishing the tumor in organ or in spheroid culture, rather spheroids are forming. So to see the effect of targeting both Notch pathway along with the cisplatin, then we treated spheroid with both this compound, and you can see there that in both primary and secondary sphere culture there is kind of drastic effect. The cells are getting killed, and there is no sphere as a significant sphere formation for both this LOI and RO inhibitors. And the viability results is also showing that in combination modes. So combination mode means where there is Notch inhibitor, either LOI and RO along with the cisplatin. So that means there is significant reduction. So in this column where is the double plus signs is there, that means LOI and RO, LI, RO along with the cisplatin is there. So there is significant reduction in viability for throughout these three OSCC cell lines. So that means if we target this Notch pathway by employing Notch pathway inhibitors, LOI and RO, along with the cisplatin treatment, then it can significantly reduce the spheroid culture by targeting the stemness, by inhibiting the Notch pathway. Going further, like as we are seeing that cisplatin, which is a major drug, is showing cancer cells are showing resistance to that. So then we tried to find out or screen some novel compounds. So for that purpose, we have collaborated with 𝒂𝒅𝒂𝒂𝒂𝒂𝒂 Chemistry Department with Dr. Arindam Mukherjee. and then we try to find out the novel compounds, basically ruthenium and iridium. These are the rare metal-based compounds, and the mesalazine derivatives. Mesalazine is a compound. Along with that, they tried to put this rare metal as a center molecules. So this M kind of representing this either ruthenium and iridium. So then we tried to find out this the efficiency of these compounds to target cancer stem cells. So for that purpose, we have taken help of our spheroid culture model, and as you can see, compared to the control, the CDTP is the cisplatin. So as we already know from my previous slides, that cisplatin is not effective in killing cancer stem cells. Then we employed this compound on 2, 3, 8, 12, and 13. These are the compounds generated by ISR Kolkata Chemistry Department from Dr. Arindam Mukherjee's group. And you can see that specifically compound on 2 and 13 are basically showing some effective or drastic result, or kind of robust result by targeting cancer stem cells. So you can compare the growth or like size of this particular spheroid compared to the other treatment condition. So to see the changes at the gene level also, we tried to do RT-PCR, but before that, this sphere diameter results is also showing that compound 1 and 2 there is significant reduction. So these are the three plots. You can see there is in compound 1, 2, and 13 there is significant reduction in sphere diameter, as well as at the viability level, compound 1, 2, and 13 are showing significant reduction of viability of the sphere culture cells. So to see the further changes at the mRNA level, we have done RT-PCR. So for RT-PCR, we have collected RNA from those treatment conditions, and then we have done RT-PCR basically. So to see changes at the mRNA level. So this color coding are the treatment conditions. So basically this brown and this green color is signifying this compound 1 and 2 throughout this all the bar graphs, and throughout this bar graphs we are showing the expression of LDH on ABCG2. ABCG2 is a drug efflux molecule. LDH on is the aldehyde dehydrogenase, that is known stemness regulator. SOX2, SOX9, KLA4, OCT4. These are the known stemness regulator factors. Notch1, Hesh1, CMYC, and these are the notch pathway related genes, inverkin and ck these are the differentiation markers. So the concept, there are two concepts, self-renewal and differentiation. So cancer stem cells basically maintain both this concept. So in either tissues or in cancer, basically cancer stem cell can self-renew and maintain its own population, and at the same time it can differentiate and generate other cells type of a tissue in normal condition, and in cancer the other cells or tumor cells. So it is showing that due to this compound 1 and 2 treatment, there is significant reduction in both like stemness regulator genes as well as notch genes, notch1, H1, CMYC. But at the same time, there is upregulation for differentiation marker, involucrin and CK10. You can see in these two conditions, for both these two genes, there is upregulation for at the mRNA level. So that means that if we targeting those spheroid with these compounds, there is notch pathway inhibition. At the same time, there is upregulation for differentiation marker. So then we got the concept that these compounds are basically maybe targeting cancer stem cells by promoting differentiation, and at the same time inhibiting the stemness. So to try these compounds more robustly, we have taken help of our notch clone, where as we are seeing that notch pathway is basically acting for cisplatin resistance. So to check the scenario in this condition, we have taken help of HICNGFP, that is continuous notch active cells. So we have plated these cells, and then again we tried to inhibit the those spheroid by targeting this compound 1 and 2. So there is also, it is also showing that this compound 1 and 2 are basically equally effective, whether it is notch continuous notch active or inactive. So that means these compounds are equally effective of targeting cancer stem cell if they are maintaining continuous notch high state. Uh, so you can see this tree plots data. There is showing that the 72-hour treatment of this compound on and 2 at 2 micromolar, sorry, at 2 micromolar dose, there is significant reduction of in both P length and HIC, and significant reduction of sphere diameter. So basically, spheres are getting shrinked due to this dying of the cells at periphery, and eventually they are dying. And at the same time, we try to find out gene expression of the already known stemness regulator, as I am already telling many times, like SOX2, KLF4, and ABCG2, as well as these NOTCH genes are getting significantly downregulated due to this treatment. And at the same time, involucrin and CK10, these are the genes marker for differentiation, so those are getting upregulated. So that means that these compounds are basically targeting stemness by inducing more differentiation. So this is the overall concept of my this part of my work, where I am showing that due to this compound, particular compound, novel compound treatment, there is reduction for stemness regulator genes, and at the same time there is regulation for differentiation marker. So that means these drugs are targeting oral cancer stem cells by inducing more differentiation. So that we have already at the stage of publishing our result. So conclusion so far are the upregulation of Notch pathway genes in cell culture of oral cancer cells signified its role in maintaining cancer stem cells. And cisplatin induced Notch high state of stemness. That means that Notch pathway is responsible for cisplatin resistance in oral cancer. And we can target these stem-like cancer cells by inhibiting Notch pathway. And at the same time, we tried to find out some novel compounds which can show its efficiency irrespective of Notch pathway activity. So for that purpose, novel ruthenium compounds, mesylazine-based ruthenium compounds are showing some very good results, and therapeutically it's more promising. Coming back to my second part of my work, so that means that as we are saying that notch, there are both notch active and inactive cancer stem cells. So to find out the factors or pathway in notch inactive conditions of stem like cancer cells, so for that purpose we have done RNA sequencing. So we have taken cells from both notch active and inactive condition, and then we have done RNA sequencing method to see that what are the changes at the mRNA level in notch inactive condition. So from that condition, we have analyzed the data from RNA sequencing, and we are showing that there is a significant upregulation for some particular signaling pathway. Basically, there are few of inflammation and innate immune response pathways, and along with that, there is a upregulation for JAK-STAT signaling pathway. So JAK-STAT signaling pathway is basically works in a inflammatory condition. Basically, they are activated by interleukin molecules in response to particular inflammation or like stress. So that pathway got upregulated in notch inactive stem like cancer cells. So we have chosen, then we have chosen this particular pathway. So why we have chosen, so there are other pathway of regulation also, but we are basically trying to find out the method or the process by which we can target cancer stem cells. And this pathway, targeting this pathway seems very much therapeutically promising, as it works in a signaling cascade manner. So it could be a good pathway to work on to basically see for a particular drugs or combination of drugs. So for that purpose, we have taken the pathway, and we then again to validate our RNA sequencing results. The result we got, is it like valid result or like any artifacts or anything? So for that purpose, we have again went back to genomics data, already available genomics data, and it is like reliably we have seen that notch pathway and JAK-STAT pathway at the genomic level at oral cancer patients are working in an inverse manner. Inverse manner means when notch pathway is active, there is low JAK-STAT activity, but in absence of notch activity, there is a high JAK-STAT activity. So that means that in absence of notch pathway, JAK-STAT pathway is basically taking care of stem-like cancer cells. So to see at the protein level that as this pathway, like at the downstream of JAK-STAT pathway, there is phosphorylation of STAT molecules, and then the signaling cascade basically activates. So to see the changes at the protein level, the phosphorylation level of basically STAT molecules, we have done Western blot, and there we are showing that in absence of notch activity by targeting by treating with LO and RO, there is significant upregulation of phosphorylated STAT molecules. So there is phosphorylated STAT1, phosphorylated STAT3, phosphorylated STAT4. But at the original level, there is no significant reduction or upregulation. So that means that in absence of notch pathway, these STAT molecules are getting phosphorylated, and overall notch JAK-STAT activity is getting high. So that means our RNS results is kind of showing the clue that inactive of notch pathway is leading to a higher JAK-STAT level at stem cells, at maintaining stem cells. So as we got these two pathway, and they are working in a more like helping each other and to maintaining cancer stem cells. And as we know that cancer cells stem cells are the like they are very plastic in nature, they are very dynamic in nature. So whenever there will be treatment, they basically change their state. They can adapt a particular pathway to survive, to skip the therapy, and again when the therapy is not there, they can come back and form the whole tumor. So from that perspective, we have taken the synthetic lethal approach. That means that two genetic factors kind of working in a synergistic manner. So if one is kind of off, the other is taking care of the whole process. But if we target both these factors or genetic factors, then the overall process has been it will collapse. So from that is the concept of synthetic lethality. So in case of spheroid culture also, we have taken help of these small molecule inhibitors for both this pathway. That includes LOI and RO for notch specific inhibition, and tofacitinib and tofa and ruxo are basically inhibitors for JAK-STAT pathway. So as you can see there, whenever there is double plus signs in these columns, so there is significant reduction in sphere formation assay. So that means that if we target both this notch pathway and JAK-STAT pathway by employing these small molecule inhibitors, there is significant reduction and drastic killing of spheroid cancer stem cell in spheroid cultures. And similar type of result has been observed. The pattern in stable clone result is also showing that in continuous notch inactive condition, that is BHAS, if we target JAK-STAT pathway by tofa and ruxo, by treating with tofa and ruxo, then there is significant reduction in sphere size diameter. So these tree plots for both this GBC02 and ACC070 cell line is showing the similar pattern of result. And at the same time, these are the result with small molecule and stable clone cell lines. Then we tried to find out the effect at by employing siRNA mediated transient knockdown of STAT3 and STAT4. So this is not continuous notch high state. So there is targeting STAT3 and 4 is not showing any result. But if we are inhibiting STAT3 and 4 by siRNA in continuous notch inactive cells, then there is significant reduction of sphere forming ability. So that means that the targeting both these pathways. So in BHAS, notch pathway is already taken care or inhibited. So then if we target STAT3 and STAT4 by siRNA, there is significant reduction. So that means this targeting both this pathway in a synergistic or in a combinatorial mode will be more therapeutically promising, and it is giving more significant result compared to their alone treatment. So the result has been further validated in a zebrafish embryo model, where we injected the cells after from this continuous notch high and continuous notch low cells by pre-treating them with ruxo and tofa. These are JAK inhibitors. So as you can see, so in control there is a huge tumor. You can see in zebrafish model also, but we targeting with ruxo and tofa is kind of inhibiting the tumor to form, and eventually the survivability plot from the zebrafish embryos are also showing that this violet line is showing BH, only BH condition. That means this aggressive tumor condition. But in case of BH treated with Ruxo and BH treated with TOFA, this orange and this black line is showing greater. So these lines are situated in the graph in a higher location. That means that in those conditions, zebrafish embryos can survive, because we are already treating the Notch pathway as well as this JAK-STAT pathway. So those zebrafish embryo tumor are basically dying. So zebrafish kind of surviving, or they're alive. So conclusions so far are that JAK-STAT signaling pathway is responsible for maintaining the Notch inactive state of oral cancer cells. JAK-STAT signaling is involved in vivo tumorigenicity for Notch inactive oral cancer stem cells, and thus targeting JAK-STATs in Notch inactive oral cancer stem cells could be therapeutically more promising. So my lab is like they are working further to say it, and we are very hopeful that it can be like more promising in more promising result can be eventually we'll find out. So we eventually published our work recently. So the this graph uh, I kept to show the overall theme of my work. So I have worked on cisplatin resistance and showing that Notch signaling could be the reason behind cisplatin resistance. So targeting Notch pathway to overcome cisplatin resistance could be therapeutically more effective. At the same time, we have seen some novel compounds from ruthenium and iridium metal-based from the mesylagine derivatives state, and those are also showing some good promising results. At the same time, the second part is of my project that was focused to find out the concept of synthetic lethality, where we found out that in absence of Notch pathway, means whenever Notch is low, there is upregulation for JAK-STAT signaling pathway in case of stem-like cancer cells for oral cancer. So targeting both this pathway could be therapeutically more promising to target oral cancer patients in future. So these are my publication throughout my PhD journey. Uh, and This was the team. So I acknowledge my supervisor, Dr. Sandeep Singh. He's the associate professor at NIBMG doing work in oral cancer. Professor Sagar Sengupta, current director of NIBMG, Department of Biotechnology, for providing me the fellowships, and Department of Biotechnology, Calcutta University, where I have been registered for my PhD program. My lab mates, Paramita Mitro, my seniors, Ankit Kumar Patel, 𝒂𝒏 and all of my lab mates like Subhashis Ghosh, Rimpa, and all have like helped throughout my journey. They're very supportive, and the lab environment is very good. So and overall research ecosystem in NIBMG is kind of very flourishing, and it is like having all kind of cutting edge technologies, NGS, mass spec, confocal. So it is a very good research institute, giving you a kind of international flavor. So yeah, I am very thankful and grateful to be a part of this particular institute in West Bengal at Kalyani. Thank you. Thank you so much.
Okay. Thank you, Subhashis, for your wonderful talk. So we have few questions. Some students and other people have also asked
Sure,
questions. So I'm going on the question. Okay.
sure,
So first of all, I'm showing that in the screen. Okay.
sure, sure.
Here in the screen, you see that. Can you see the question?
How do we scale this to a mass level? Yeah, I can see the question. Anirnay Basu. Okay, so yeah, so for this, it's not like important to see initially see at the mass level. So these results are basically some initial hits. So to see it further, so as I have already shown in vivo result but for as a preclinical result, we have to do in more like mice model. Then there will be collaboratively we can collaborate with some companies like drug companies to design a particular kind of compounds. And this there is kind of clinical research setup where there is like stage wise or step wise there are processes. So for that bigger good, we have to collaborate with some clinical setup, as well as there will be some pharma companies. So these are the overall picture of developing some particular organoid particular drug or inhibitors. So in that sense, these are the small contribution initially, but eventually if we work on these particular discoveries, then we can do something really good.
Okay. Then there is another question from Ankana Bhattacharya.
Yes, yeah, Ankona, yeah, it's very good question. So yes, breast cancer is somewhat obviously it is genetic. There is genes called BRCA1 and 2. These are kind of known genes, and these are like tumor suppressor genes. So, and these genes can be transmitted from the mutation can be transmitted from mothers to like eventually offspring, so siblings. But the thing is, there is as you know, like cancer is a multifactorial disease, so only genetic mutations will not develop the cancer, but there is chance. And if there is mutations like on mutations is already from the mother, so those people will be kind of predisposed to the particular cancer, so there will be high chance. So eventually there is kind of two hit theory. So if there is two copy of a particular gene in our body, as we know. So if one gene is already mutated, so those people are predisposed condition to that particular disease, cancer. And again, if there is mutation for the second copy, then the cancer will eventually develop in those people. So yeah, cancer is a kind of genetic disease.
Okay. Then there is a question from Saif Alam.
Yes, can we reduce the cancer rate of common thread? Yeah, say follow. Yeah, so there is researchers throughout the globe, as I have already shown the hallmarks of cancers. So cancer cells have some basic characteristics that is kind of common between throughout the cancers. So there are multiple cancer, the multiple organs we have, there are cancers for those particular organs. But yes, throughout the cancers, different tissue cancer, the cancer cells basically they retain some common features. So that's why it's kind of helpful for us to summarize the overall cancer cell features, so that we can work on them, and we can then eventually we can try to discover something about the unknown aspect of those features, and then we can eventually research and we can target those features. So yes, there are some common traits for cancers.
Okay. We have another question from Ananadatta.
Consistent in that sense, I can say that notch on notch pathway receptors, that includes notch on, those mutations are acting as a kind of divergent mutations from the data, we can say. So yes, in Indian context, notch on mutation is kind of consistent, you can say, and it is kind of working as a divergent mutation for overall tumor oral cancer initiation and progressions. Yes.
Okay. So we have another question from Monojit Paul.
ওকে, মনোজিৎ পাল, হ্যাঁ, আলসারেটিভ কোলাইটিস এটাও একটা স্টেজ হিসেবে কাজ করে। সো ইনিশিয়ালি আলসারেটিভ কোলাইটিস থাকলে যে সেটা ইভেনচুয়ালি ক্যান্সার হবে সেটা না, যদি আমরা আর্লি স্টেজের ডিজিজটার ট্রিট করে ফেলি, বা জিনিসটা আমরা প্রপার মেডিসিন নিয়ে সেটাকে ট্রিট করতে থাকি, তাহলে সেটা অবভিয়াসলি ইভেনচুয়ালি ক্যান্সার হবে না। কিন্তু সেই রিস্কটা থেকেই যাবে যদি আমরা ওটা সবসময় ওই ইনফ্লেমেশনটা থেকে যায়। কাইন্ড অফ ক্যান্সার স্টার্টই হয় ইনফ্লেমেশন, ওভারঅল একটা টিস্যুর ইনফ্লেমেশন, ওইসব একটা জেনারেল ক্যারেক্টারিস্টিক্স হিসেবে কাজ করে। সো আলসারেটিভ কোলাইটিসও কাইন্ড অফ বলতে পারো যে গাটের ওটা কাইন্ড অফ কন্টিনিউয়াস ইনফ্লামেশন। সো যদি সেটাকে আমরা সময়ের মধ্যে ট্রিট করতে পারি, তাহলে ক্যান্সার হওয়ার চান্সটা অনেকটাই কমে যাবে।
ওকে
এর উপর হ্যাঁ, এটা একটা বাইরেও কোলন ক্যান্সার বাইরেও একটা খুব ইভেন আমি এখন ইউএসএ তে আছি, সো এখানেও এটা অনেকটা বড় ক্যান্সার, লাইক এটা খুব কমন এখানেও। তো এর উপরে রিসার্চ অনেকটাই হচ্ছে, এবং থ্রু আউট বড় বড় ইনস্টিটিউটে রিসার্চ ফান্ডিং পেয়ে অনেকে এর উপরে কাজ করছে, এবং অনেকটাই কাজ হয়েছে এর উপরে।
Okay. Next question from Shubhajit Pal.
Yes, very good question actually. That's why I'm smiling. So yeah, so as you know, like microbiome is really a hot topic nowadays, and it has that value to look after, because we know that the current research is showing that in our body, rather than like our own cells, there are more of this microbiome or bacterial cells are there. So that means that they have some really important role in overall our health and disease. So yes, in case of oral cancer, that is also true. And basically oral microbiome is playing some important role in oral tumor initiation and progression, basically at the progression level. So research is also going on oral microbiome to see its important role in overall tumor progression and overall therapeutic response. So yeah, it is very important, not only for oral tumor, for other cancer also. The microbiome is playing some important role nowadays.
Okay. We have another question from Shorajit Majumdar.
Mhm. Yes, Ishwarjit. So, as I have already shown that for cancer initiation, there is some important role for genetic instability and mutations, inactivation, gene activation. Those are playing important role at the cancer initiation stage. But eventually, when the kind of cell getting altered genomically, then they will try to escape the overall control mechanism. They will try to proliferate. So at those stages, the other factors, that is basically non-genetic changes, like it can be phenotypic plasticity, it can be non-genetic epigenetics changes, as well as tumor microenvironment, the vicinity of a particular tumor. Those factors are really, really important for metastasis and overall tumor progression. And at the same time, at the therapeutic perspective also, those factors are really important to look after.
Okay. So we have another question from Ananya Basu.
Where such rare metals used in study has got some with the chemical nature or tonic. Yeah, so these compounds are basically not so we cannot include any kind of compounds. So these rare metals are kind of from the toxicity perspective as well as from other perspective, it has been already tested throughout those experiments and overall the lab protocols they have already. So throughout this process, they're showing basically these compounds are more kind of stable, and it has that potential to be not so toxic to normal cell, but at the same time they can be more effective in targeting oral cancer stem cells. So from that perspective, we have taken those compounds. It's nothing to do with some other things, but the more scientific parameters are basically playing some important role for choosing those compounds.
She has one back-to-back questions. I think
Hmm.
this one is also addressed here.
Yes. So as I already told Orinia, that there are compounds basically are basically chosen from their chemical nature, their bonding, how they will be in aqueous solution, and how they will go into a particular cell. So from that perspective, people are basically choosing these compounds. So it's same for any kind of drug compounds or conjugates. And specifically, we have just taken the rare compounds ruthenium and iridium, but we tried to conjugate or put those metallic compounds in mesalazine. Mesalazine is kind of already known compounds that are basically already in drug markets, and people are using that for some inflammatory disease, inflammatory bowel disease, those things. So yeah, it's kind of, our compounds are conjugate one, so it has been taken from safety, cytotoxicity, and other scientific, considering other scientific parameters.
Okay. As one more thing, as I have IBD, so I don't know about this compound because my medicines contain this kind of compound. So I'm a
Okay.
patient already. So the medicines which I used to take, those have
Oh,
this kind of in the configuration in the
yeah.
components. Okay.
Yes.
So we have another question from Shunali Mukherjee.
Okay. And the radio is is there any molecular approach to detection? সোনালী মুখার্জি, হ্যাঁ, এটা করা যেতে পারে। সেক্ষেত্রে দেখুন, রেডিও রেজিস্ট্যান্স বা এগুলো যখন স্টার্ট হয়, বেসিক্যালি আমরা যখন ট্রিটমেন্ট, কোনো একটা ট্রিটমেন্ট স্টার্ট করি, সেটা রেডিও হতে পারে বা কেমো হতে পারে, তারপর সেই সেলগুলো তার এগেনস্টে রেজিস্ট্যান্স দেখায়। আমরা প্রথমে ভাবি কি ডিজিজটা হয়তো ঠিক হয়ে গেছে, টিউমার কমে এসছে, কিন্তু কি হয় যে, যেটা আমি বললাম আমার প্রেজেন্টেশনে, যে কিছু সেল আছে যেগুলোকে আমরা ক্যান্সার স্টেম সেল বলছি। স্টেম সেল কি করে, বিভিন্ন যে থেরাপিউটিক আমরা স্ট্রেস দিই, যেমন এটা কেমো বা রেডিও হতে পারে, তারা নিজেদের মতো করে নিজেদের ডিএনএ বা নিজের সেলের মধ্যে যে সমস্ত মেকানিজম আছে, সেটাকে তারা এমনভাবে চেঞ্জ করে যাতে তারা ওই যে স্ট্রেস কেমো বা রেডিও, সেটা থেকে ওরা বেঁচে যায়, এবং তারপরে আবার ওরা ফিরে এসে টিউমার ফর্ম করে। সো ওই মেকানিজমটা ধরার জন্যই আমি যেটা বললাম, প্রিসিশন মেডিসিন ওইভাবে করা হচ্ছে, যাতে কি প্রত্যেকটা পেশেন্ট হচ্ছে আলাদা, এইভাবে আমাদের ভাবতে হবে। তার সেলগুলো, ক্যান্সার সেলগুলোও আলাদা, এবং ক্যান্সার সেলগুলোর মধ্যে যে চেঞ্জেসগুলো হবে সেটাও আলাদা। সো প্রত্যেকটা পেশেন্টের ক্ষেত্রে সেই আমাদের সেলগুলোকে নিয়ে প্রত্যেকটা পেশেন্টের যেগুলো মলিকুলার চেঞ্জ, মানে ডিএনএ, প্রোটিন বা আরএনএ লেভেলে, সেটা যদি আমরা টেস্ট করতে পারি, সেটাই এখনকার কনসেপ্ট, সেটাই যে পার্সোনালাইজ বা প্রেসিশন মেডিসিন অ্যাকচুয়ালি সেভাবে কাজ করছে, যাতে প্রত্যেকটা পেশেন্টকে আমরা ইউনিকলি ভাবি, এবং তার যে চেঞ্জেস হচ্ছে ডিএনএ, আরএনএ বা প্রোটিন লেভেলে, সেইভাবে আমরা ট্রিটমেন্টটা দিতে পারি। সো এই জিনিসগুলো অনেকটাই এখনো, মানে আমাদের দেশেও এসেছে সেটা, কিন্তু অনেকটাই সবার কাছে হয়তো অ্যাভেলেবেল হয় না, ডিউ টু সাম ফিনান্সিয়াল অ্যান্ড আদার আসপেক্টস। বাট এটা ইভেনচুয়ালি পপুলার হচ্ছে, এবং লোকজন জিনিসটাও অনেকটাই কম দামের মধ্যে, কম ইসের মধ্যে আসায়, খরচের মধ্যে আসায় লোকজন এটাকেও ধীরে ধীরে করতে পারবে ইন ফিউচার।
Acha. We have another question from Ankana Bhattacharya.
Okay. After treatment of cancer, in many cases, the cancer again arises. Why? And if they have been destroyed from molecular level. So, Ankona, as I told already, there is particular cell called cancer stem cells. So, yes, so I have already, in previous question also, I have trying to say that thing, that there is cancer stem cell, basically those cells are basically responsible for coming back of tumors. So rather than targeting the other cell population, that's why we are more focused to target cancer stem cells, because if we can target these cancer stem cells, then we can totally diminish the tumor. So but in that process also, cancer stem cells are so smart that they are showing some kind of mechanism or others that are basically giving them the power to skip drugs. But people throughout the world are basically really trying hard, researcher, to find out the mechanism for each and specific cancers, the stem cell mechanism or the other factors, those are basically conferring those resistance. So yeah, future is kind of bright. People are really working hard to find out the mechanism.
Okay, we have another question from Akash Vishwas.
What are the kind of epigenetical factors that can increase the risk of oral cancer? Are there any determined set of factors with respect to epigenetics? Yes, Akash, there are epigenetic mechanism, but epigenetic comes when there is so these factors are basically interrelated to each other. So there will be events, initially there will be genetic events, genetic insult that includes mutation, copy number alteration. Those are the factors for cancer initiation. Eventually epigenetics factors will come, and the overall tumor microenvironment. So saying about epigenetics, there are particular histone proteins as well as epigenetic modification of DNA. So those are very unique, as I am trying to say that in each cancer and in each patient, at the cellular level also, the things are very complex. Means there is not a particular hit, particular on target to hit. Rather, we are trying to find out the factors which are basically interacting with each other, and then they are ultimately that aggressive tumors is behaving. So yes, there are epigenetics factors at the DNA level, CpG island, those things, methylation, DNA methylation. Those are the epigenetic modification of DNA, as well as histone modification. Histone modification includes acetylation, methylation, those things. So yeah, this is the kind of context. The context is very complex, but yeah, people are trying really hard to find out those factors important for specific cancers.
Okay, we have a long question right now from 𝑦𝒏𝒕𝒆𝒏𝒂𝒕𝒊𝒐𝒏.
Okay, rhetoric, yeah, as the Jackson. The things. Yes, rhetoric basically genes. As I'm telling that precision medicine concept, so we are basically so at the therapy level, you can say that we have to screen the oral cancer patient. So suppose we find out that in a group of patient there is mutation for notch-on. So then we can think that in those patients we can target the oral cancer cells by targeting JAK-STAT pathway. But if there is like not notch on mutation in those patients, then this therapy we have to like think about some other methods. So in that perspective, this mutational signature will give us the like pretext or like pre-knowledge to how to treat a particular patients based on this notch on mutation is present or not. So these things are very specific. And for your second question, are there that activate notch across. Yes, as you like already mentioned that single cell approach. So yes, sequencing patient tumor derived cells at the single cellular level will definitely give us a more clearer picture to see that at the mRNA level what are the changes of these genes at for a particular pathway. Either it would be Notch or JAK-STAT or some other pathways that are important for oral cancer or some other cancer. Then only we can as a downstream or as a therapeutic aspect, we can think about some therapy or inhibitors. So yes, these things are very specific for each patient at the cellular level also. So single cell approach is definitely a very good on to find out the changes at the mRNA level for a single cell.
Okay, we have another question from 𝑰𝒏𝒕𝒆𝒏𝒂𝒕𝒊𝒐𝒏.
Hmm. Yes, I will the study of the change in the genome.
And he has a following up question next. I'm showing you that also.
Yes, so talking about the previous question, yes, there is always, yeah, you are right. There is always a chance to like diagnose early. So for that purpose, also like pharma companies, they are trying to develop some screening kit, basically in screening kit in that sense that in from the your body fluid, specifically from blood, people like they can collect your DNA from the blood sample, like that is called fluid biopsy, and then from that circulating that is called circulating DNA in the blood. So from that molecule also people can see that changes at that genome level, the mutation they can see. They can also see the methylation steps. So these are the process or research topic people are working on to find out the diagnosis more relevant and more accurate, to see the changes at the molecular level and then to decide the therapy based on those changes. And your second question is there a crystal structure the protein? So yeah, for second question at the crystal level, so for that purpose also there is collaboration is needed. So these things are not easy to do. So for that purpose, in biomedical, specifically in biomedical research, it is very important to collaborate with the relevant group and to do further studies. So yeah, that is very relevant on to see the protein, the structural changes at the protein, specifically notch-on and H1. So those things will eventually help us to develop some particular compound or drugs that can detect those changes at the structural level, and then it will be more accurate to target those pathway. And yes, ABC transporter is the transporter basically responsible for drug efflux or influx. So it has been shown that in case of stem cell population, this particular ABCG2 expression is kind of high, or it is maintaining its expression by doing the effluxing more drug compounds from the cells. So it is a mechanism of cancer stem cells to become resistant to a particular drug. So ABCG2 is kind of drug transport. It works as a drug efflux molecules for oral cancer stem cells and some other cancer also.
Okay, we have another question from Shucheta Malakar.
Okay. Tumors which have undergone metastasis are metabolically different from normal cells, rapidly growing cells, so nutrient availability is less. So what are the main changes? Yeah, Sucheta, these the cells basically which metastasize to some other part. So it has been shown throughout recent research is also showing that there is concept of seed and soil concept. So that means that if some cells from its primary site metastasize to some other part, it depends that where it is going, and what is the location and surrounding where the cells are getting attached and then eventually form tumors. Metabolic changes is also important, but that for metastasis the cells are becoming more motile. They are getting some characteristics that helps them to basically run into some other place or go into some other place, and then eventually they form a colony there. And overall these metabolic changes are very dynamic. So metabolomics of cancer cells are really a good topic to work on, and people are also working in this particular topic to find out the plasticity at the metabolic level and how we can target those plasticity to get rid of any kind of cancers. So people are really working hard on that topic also.
Okay, we have another question from Sonali Mukherjee.
Is the gum infection related with oral cancer? Is oral hygiene important? Yes, madam. So certainly, ma'am. So yeah, gum infection is kind of it can act as a factor, but it depends what kind of infection and what type of factors are there. So that is like a kind of complex context. There could be many factors. So but yes, overall oral hygiene is really important to maintain, because cancer is a disease, from my knowledge I have seen that continuous inflammation means the things are getting there is kind of continuous stress. If you are not taking care of your oral hygiene, smoking, regular alcohol consumption, and other tobacco consuming, these are kind of very big risk factors for oral cancer to develop. So to taking care of those factors will minimize your chance to getting cancers. Yes. So in that sense, oral hygiene is really important to maintain.
Okay. We have another question from Sohaili Basurai.
Okay, I have a question regarding immunotherapy. Nowadays, CARs therapies and bispecifics are used as separate treatment, but in near future, what are the prospect of using CAR therapy, and also what are the prospect of development of tri-specific antibodies in case of solid tumor? Yeah, Shaili, these questions are very intriguing and very important questions to look after in future, and people are also doing research on this particular field. And immunotherapy is the therapy which are like emerging as kind of major tool for us to target any kind of advanced tumor. So in that perspective, as you mentioned, these bispecifics and trispecific antibodies are playing some important role, and people are doing research on these topics. So eventually, in future, we may say we may see the therapeutic approach from the immunotherapy perspective, and companies are developing such drugs. So it will be really important for future therapy against any cancer, specifically in oral cancer also.
Okay. Another question is from Sritama Sarkar.
Notification. Thank you, Sitama. In Notch inactive cancer stem cell is only JAK-STAT pathways and upregulated, or any other pathways are also involved. Many interplay according in a cell. Yes, I'll first say your second part. Yes, at the cellular level, there are many signalings, and interplaying between them is the gist, basically giving the cells to drive or like to be alive. So from the normal cellular biology perspective, yes, we know there are many signaling pathways that interact with each other, and that is basically important to for cell survival. But yes, for oral cancer stem cells, as I am telling many times that our approach was to see at a particular patient what are the changes at the notch pathway, and how this mutation in notch pathway are giving this aggressive oral cancer outcome. So for that purpose, we have sequenced the oral cancer stem cells in from the notch inactive conditions of stem cells, and then we try to find out by doing RNA sequencing what are the changes at MR level, and we find out that JAK-STAT pathway is coming out as a major upregulation in notch inactive condition. So we choose JAK-STAT pathway basically because this pathway, as first of all, it is a signaling pathway. That means it has some downstream molecules also that ultimately goes into the nucleus and doing some changes at the transcriptomic levels, at the translation level eventually. So for that perspective, from the therapeutic angle, we try to include this JAK-STAT pathway, because if we can target these pathways, then it will be more conveniently we can target cancer cells. So from that perspective, we have taken JAK-STAT pathway.
Okay. Another question from Akash Bishas.
Cause is telling can change in climate, temperature, food habits, lifestyle of the population directly affect the rate change of mutation from B19. But so factors slow down the rate of. No, Akash, these things are not working in that sense, I think. So rather, this obviously you are right that these are the factors, basically climate, temperature, food habits, lifestyle. These are the factors. So wherever we live, it is all about like surroundings, in which environment we are living. It could be environmental stress. It could be like the pollution, environmental stress, the pollution, the food habit, the climate. So these things are obviously affecting us, but as a food habit also, in a food habit sense, I can say that our traditional food habits, whatever we had, like in India, so that things are like it kind of healthy, you can say, but eventually we adapted the Western kind of food culture. So these are the changes we are doing in our lifestyle. So these things kind of making it risk factors. the lifestyle and food habits for eventually developing cancers. So these things really matters in a long term manner. But uh, but don't have some like specific kind of correlation from benign tumor to like metastasis. Those are very specific things happening, including all the other factors.
Okay, we have another question from Subhashis Ghosh.
Okay. Like DNA methylation pattern or modification differ between various types of colon cancer. Subtypes. Yeah, very good question and very intriguing question from Snehasis. So basically, yes, apart from the genetic changes, so genetic changes are important for cancer initiation. But as I already told, that eventually tumor will develop, and as it is showing therapy resistance, so the current research is showing that epigenetic factors, specifically DNA methylation, histone modifications by either acetylation, methylation, and some other ubiquitination. So these are the factors or basically epigenetic signature that is playing some important role for personalized medicine also. So as each patient has its unique genetic signature, the patient also has its epigenetic signature, so and also has its microenvironmental signature. So as a from the cancer perspective, cancer is a very complex disease, you can say. So all this taking care of all these factors and getting at least getting knowledge from all this perspective is really helpful to target a particular cancer in a particular or in a specific patients. So yes, the subtype and overall epigenetic knowledge will definitely help to get better treatment for a particular patient.
Okay, we have two more questions. So taking it fast, actually, you are engaged right now. It's almost 90 minutes. So
Mm,
it's
it's okay.
already morning. I think you have to go to lab, I guess. So
Yes, yes, I can. Yeah, it's very entertaining, like interacting. So yeah, I'm also enjoying the
okay,
things.
okay. If you are fine, then it's okay. So I'm just
Yeah,
worried
yeah,
about
yeah.
your.
These are very interacting. So yeah, it will actually eventually help me also to see the other perspective also. So can a person
Okay, okay. So
stage
here is the question from 𝑦𝒏𝒕𝒆𝒏𝒂𝒕𝒊𝒐𝒏.
of genetic disease like skid have
And there is a follow-up question along with it.
been told.
Is there any particular step to prevent this? So I'm showing the
Okay,
first
okay.
one.
Yeah, it is like skid have a chance. Treatment of genetic disease like skid cancer due to inhibitor like 𝑉53 in the recipients. Yeah, yeah. I use these questions are very specific questions you are asking, very good questions. But yes, so for any kind of disease, first we have to see the what are the changes at the molecular level, specifically like at the sequencing, by doing sequencing at the DNA level, at the mRNA level, and eventually at the protein level. So combining these methods is the kind of thing we have to do in future. So from that perspective, yes, for diagnosis and like early detection, genetic mutation is important. But eventually for treatment, we can see the changes at the cellular level. And talking about cancer stem cell, so we can see changes at the stem cell level. And based on the alteration at the stem cell level, we can target those specific pathway. Like in my case, I have shown that targeting both notch and JAK-STAT pathway could be more therapeutically promising. So so in that sense, it is important to first see the genetic or molecular changes, and then decide the therapy. So yeah, and protein control, protein P53, these are like all related. So eventually P53, for personalized medicine, I have shown that there is a particular group of patient where TP53, the gene of the P53 protein, is mutated. So in those cases, like targeting P53 mediated mechanism is kind of important for better efficiency. So yeah, these things are very specific, not in general. We can say that each drug will work for every patient. It's not like that.
Okay, so we have another question from Shomodip Pal.
Okay. So Mudip ask like, is there a universal set of microenvironmental conditions that promote the development of a single pre-malignant cell to a to begin its cycle of uncontrolled cell division? How can we totally cure a disease that is continuously evolving? Is it really possible? So for your first question, Shomodipto, so yes, so microenvironment factors we can see in that sense that cancer is kind of following the path of evolution. So the cancer cells, when it starts and then eventually try to adapt with its environment, and then the environment also kind of interact with those cells, and eventually it is like evolution of that ecosystem. And in that ecosystem, cancer cells are there as well as tumor microenvironment factors are there. So all of them are trying to give a particular advantage to cancer cells to survive, and then eventually grow and form the tumor, as well as they become metastasized to other organs, other healthy organs. So yes, it is a kind of overall complex situation where multiple factors are involved, and all these factors are basically giving cancer cells to kind of advantage. And yeah, so as these tumors are evolving, so yeah, as you are asking, really good question, that is it really possible to target this evolving disease? Because it is really hard. You are right that it is kind of very hard to target. But so there are multiple factors, like early detection is one factor, and if the disease is already progressed, metastasized, and already like in advanced stage, then the treatment regime would be definitely would be different. So so as I like telling multiple times that each patient and each disease and each cancer is specific, and so we have to like see each tumor in a unique way. And for that purpose, we have to that personalized medicine or precision medicine is the thing for future. And in a more robust way, more methods are coming in to help us to see the molecular changes and how we can treat in a better way. So yeah, these things are going on.
Okay, the next one is from Shuvaloki Sinha.
Sovalokhi asked that what is the current need of the hour in cancer research? What aspect of it requires our urgent research attention? Yeah, as I already told, like we have to think in that sense that Sovalokhi, each patients and each tumor is unique, and for like there is no common drugs or no common methods that can work for each patient. So you have to see that in a particular patient at what stage the tumor is, how it has been spreaded to other parts, and what are the molecular changes at the gene level, at the protein level. Then only we can target those patients. So yeah, these things are very unique. Yeah.
Okay, another from Iqbal Sheikh.
Iqbal Sheikh asked that with the increasing availability of genomic data, how do researchers ensure the accuracy of genetic information? Yeah, it's playing some important role basically to see the genetic changes by doing these NGS methods are very common nowadays, like next generation sequencing. So these methods are basically helping us to see changes at the alteration at the DNA RNA level. So those are basically helping us to see each patient differently, and then based on those genomic landscape or genomic signature of each tumor, we can define our therapy or drug based on those signature. So it is for personalized medicine, these things are very important.
Okay. I have got one question. Someone has sent me a question through mail also. So he was unable to comment in Facebook. So he has sent me a mail. So
Okay.
his name is Pathik, asking if a cancer patient, in bracket there he has mentioned leukemia, goes to hypoxic condition for a long time, then will it be possible to cure cancer due to prolonged erythropoietin secretions?
Repeat the last part, please.
If a cancer patient, leukemia, in bracket, goes to hypoxic condition for a long time, then will it be possible to cure cancer due to prolonged erythropoietin secretions?
Prolong.
Erythropoietin secretion.
Oh,
Erythropoietin.
erythropoietin secretion. Pratik, these questions are really specific questions. So I'll it is not important to say yes or no. Rather, the patient, the overall tumor condition. It depends on the overall tumor condition. If it has some and hypoxia is a research topic. People are doing that to target to basically deprive cancer cells of oxygen, and that is a kind of therapeutic aspect people are doing on. But yeah, these questions are very specific. So in that particular patient, if that overall so erythropoietin, as you saw, those are the factors or already known proteins or secretory factors. Those are basically working. So to see the alteration at the molecular level, at the protein, or as you told, or secretory factors level, and then to see the others relevant factors, it will help us to basically develop some more specific therapy for that particular patient, so that there will be some inhibitors for erythropoietin also. So in those perspective, we have to see, not in a general term that this will work or not. It is like a more like important question to rather see in more details and see the other factors, relevant factors, and then decide what will work or not.
Okay. Um, we have now have almost has reached the end of the of this program. So এবার আমি মানে সায়েন্টিফিক ডিসকাশনের পর জাস্ট একটু যেহেতু আমরা সায়েন্স কমিউনিকেশন বা সায়েন্স পপুলারাইজেশনের জন্য আমরা নেটিভ ল্যাঙ্গুয়েজ বা লোকাল ল্যাঙ্গুয়েজে আমরা কমিউনিকেট করি। সো
Okay.
বেসিক্যালি তোমার একটু সাজেশন বা টিপস বা তোমার পরামর্শ বাচ্চাদের জন্য এবং ক্যাটেগরি মানে যারা স্কুলে আছে, যারা কলেজে আছে, যারা ইউনিভার্সিটিতে আছে, মানে প্রত্যেকের জন্য তো কিছু স্পেসিফিক থাকবে আশা করি।
হ্যাঁ, হ্যাঁ, সেই রকম আমরা আমরা 12 আসতে মানে মাধ্যমিক অবধি তো মানে সেটা পড়াটা
সবাই
অন্যভাবে থাকে ওই অবধি বলে, মানে আমি নিজেও, হ্যাঁ, আমি বেসিক্যালি হয়তো বিএসসি তে এসে বা এমএসসি তে ফাইনাল ইয়ারে এসে রিসার্চের কথাটা ভেবেছিলাম। তো ওই স্টেজে ওইসব আসে না বেসিক্যালি। বাট কেউ যারা যদি সায়েন্স নিয়ে পড়ে, কেউ ধরো বায়ো সায়েন্সে করছে, বিএসসি করছে, সো বিএসসিটা আমি বলবো যে এখন তো ইভেন অনেক রিসোর্স এসছে অনলাইন থেকে শুরু করে। তো জিনিসটা যতটা বেসিক গুলো শেখা যায়, যে বিএসসি গ্রাজুয়েটে যে কোনো সাবজেক্টই থাকুক, সেখানে তুমি চেষ্টা করো কি যতটা বেসিক নলেজ পাওয়া যায়। খুব ভালো বই পড়তে হবে, এমনি এমন না যে নোট নিয়ে এক্সাম উতরে গেলাম, সেটা না। নোট তো থাকবেই অবভিয়াসলি, দেয়ার উইল বি টিউশন, আমি নিজেও পড়েছি।
বড় প্রবলেম, শুধু নোট পড়ে যেটুকু পরীক্ষা আছে তার বাইরে
না, না,
পড়বো না।
না, এটা লং রানে হেল্প করবে না। সো আমার ব্যাচের সাথে হয়েছিল কি, লোকজন ওই টিউশন যাব, ওখান থেকে নোট দেবে, শুধু ওইটুকু পড়ে নিলাম, তারপরে এই করলাম, এইভাবে হবে না। লাইব্রেরি যেগুলো থাকে একটু চেষ্টা করো, মানে এমন না যে ওখানেই বসে থাকতে হবে সারাদিন। যতটুকু একটু ইউনিকলি নিজের মতো করে পড়া জিনিসটা, যতক্ষণ না নিজের মধ্যে আসবে তুমি।
অনার্স বা ইউনিভার্সিটি বা কলেজ সিলেবাস যেটা আছে সেটা বাইরেও আমাকে পড়াশোনা করতে হবে।
রাইট, কারেক্ট। তো যতটা করা যায় নিজের, এবং অনেক সময় ভালো লাগে যদি কোনো সেই জন্যই মানে নিজের পছন্দের সাবজেক্ট চুজ করাটা বেশি ইম্পর্টেন্ট। তাহলে সেটা পড়লেও বোরিং মানে পড়া মনে হবে না, অনেকটা গল্পের বইয়ের মতোই তুমি পড়ে যাবে। সো আমি যেমন জুলজিতে পড়তাম, আমার অনেকের ক্লাসিক্যাল যেটা জুলজি ক্লাসিফিকেশন, ওগুলো অনেকেরই ভালো লাগতো না, বাট আমার অনেকটা এগুলো ভালো লাগতো। মানে প্রথম থেকে এগুলো পড়ে তারপর আমার এমএসসি বায়োটেকে ছিল, তখন একটু রিসার্চ বিএসসির ফাইনাল ইয়ারে মনে হলো যে সবাই তো সেই ট্র্যাডিশনাল জুলজি নিয়ে, আবার এমএসসি ওইভাবেও হয়, বাট আমার একটু বায়োটেক নিয়ে পড়ার, বেসিক্যালি স্টেম সেল নিয়ে একটা কাজ করার বা পড়ার তখনই একটা ইচ্ছে হচ্ছিল, স্টেম সেলের ব্যাপারে যখন জানলাম। সো ইভেনচুয়ালি এমএসসিতে আমি বায়োটেক নিই, কল্যাণ ইউনিভার্সিটি থেকে মলিকুলার বায়োলজি অ্যান্ড বায়োটেকনোলজিতে এমএসসি হয়েছে আমার। সো সেখানে পড়ার সময় আমি যেগুলো ওভারঅল যে মলিকুলার টেকনিক্সগুলো, ট্রান্সক্রিপশন, ট্রান্সলেশন, এই যে মেথডগুলো ধীরে ধীরে জানলাম, ডিএনএ রেপ্লিকেশন, এই যে স্পেসিফিক মলিকুলার স্টেপসগুলো, ওগুলো খুব ইন্টারেস্টিং ছিল। তো জিনিসটাকে একটু নিজের মতো করে ভালো লাগলে জিনিসটা ইজি হয় কন্টিনিউ মানে পার্সু করা ফর ফিউচার। সো দেন ইভেনচুয়ালি যখন এমএসসিতে কেউ ভর্তি হবে, সে যদি চায় যে রিসার্চ ফিল্ডে আসবে, তবে তার তার জন্য আমার একটা স্পেসিফিক সাজেশন যে সে ফার্স্ট ইয়ার থেকে এমএসসির সে যদি নেটের বেসিক্যালি আমাদের দেশে আইসিএমআর, সিএসআইআর, ডিবিটি, আইএআর আছে, এগ্রিকালচারের একটা নেট আছে। সো যেই নেটগুলো আছে, ডিবিটি নেট আছে, যেমন আমার ক্ষেত্রে আমি ডিবিটি জিআরএফ কোয়ালিফাই করেছিলাম। সো একটা জিনিস করতে পারো কি তোমরা, কোশ্চেন পেপার যে লাস্ট 5 ইয়ার্স, স্পেসিফিক্যালি লাস্ট 5 ইয়ারের কোশ্চেন পেপারটা সলভ করতে থাকো ফার্স্ট ইয়ার থেকেই। তাহলে কি হবে, যদি ওই তুমি ওই কোশ্চেনটা সলভ করার সাথে সাথে যে আশেপাশে টপিকটা পড়ে নিচ্ছ, ওটা তোমাকে অনেকটাই হেল্প করবে লং রানে। ওটা তুমি বারবার ওই ফিরে গিয়ে, এবং কি কোশ্চেন প্যাটার্ন তুমি দেখবে যে অনেক রিলেটেড, মানে একই টপিক থেকে কোশ্চেন থাকে প্রত্যেকটা ইয়ারে। এমন না সেম কোশ্চেন থাকবে, বাট ওই টপিকটা তুমি যদি ওই ফার্স্ট টাইম কোশ্চেন সলভ করতে গিয়ে পড়ে রাখো, তাহলে নেক্সট টাইম তোমাকে ওটা হেল্প করবে যে কোনো কম্পিটিটিভ এক্সাম কোয়ালিফাই করার জন্য। মানে এটা একটা বড় স্টেপ আমার মনে হয়, যে প্রিভিয়াস ইয়ারে কোশ্চেন গুলো সলভ করা গিয়ে গিয়ে ওটা অনেকটা হেল্প করেছে আমাকে, নিজেকে অনেকটা হেল্প করেছে। সেই জন্য আমি তোমাদের সাথে মানে এটা বলতে পারি যে এটা খুবই ইম্পর্টেন্ট একটা স্টেপ। সো তারপর বাস, ইভেন আমাদের ওয়েস্ট বেঙ্গলেও অনেক ভালো রিসার্চ ইনস্টিটিউট আছে, ইন্ডিয়াতে আছে, বাকি অন্যান্য স্টেটে আছে, এবং কি বাইরে কারো যদি আসার ইচ্ছে থাকে তাহলে সে আসতে পারে, পিএইচডি করতে পারে। তো এই স্টেপগুলো পরপর হয়ে যাবে, কিন্তু থ্রু আউট দ্য প্রসেস একটু ধৈর্য রাখতে হবে, আর যতটা পারা যায় নিজের মতো করে জিনিসগুলোকে মানে বলে না অ্যাসিমিলেট করা, নিজের মধ্যে আত্মীকরণ করে নিতে হবে জিনিসগুলো। তাহলে কি হয় যে লং রানে তুমি জিনিসটা হয়তো মানে তোমার ইমিডিয়েট হবে না, কিছুটা ভুললেও ভুলে যাবে, কিন্তু যখন তুমি কোশ্চেন কোনো দেখবে বা কিছু দেখবে, তখন তোমার ওটা অবভিয়াসলি মাইন্ডে ওটা রিকার হতে থাকে। সো এভাবেই কিছুটা প্রিপারেশন নেওয়া যেতে পারে।
আরেকটা জিনিস জিজ্ঞেস করবো, কারণ তুমি এখন হাওয়ারের মতো জায়গায় আছো। আমাদের বাচ্চারা স্কুলে পড়াকালীন একটা সিদ্ধান্ত নেয়, আমি ভুল বা ঠিক সেটা বলবো না, কিন্তু সিদ্ধান্তটা হচ্ছে যে তাদের অনেকেরই ইচ্ছা থাকে যে আমি ডাক্তারি পড়বো বা মেডিকেল ফিল্ড মানে নিট পরীক্ষা দেবো বেসিক্যালি। তো পরে কি যে তারা ফিজিক্স, কেমিস্ট্রি আর বায়োলজি নিয়ে মানে ম্যাথামেটিক্সটাকে ইদার খুব নেগলেক্ট করে, নাহলে ফিজিক্স, কেমিস্ট্রি, বায়োলজি, ম্যাথামেটিক্স পুরো বাদ দিয়ে টুয়েলভটা পড়াশোনা করে। তারপর কিন্তু দেখা যায় যে ইভেনচুয়ালি এসে বায়োলজি কোনো একটা ফিল্ডে আসছে, এবং এটা একটা খুব ভয়ঙ্কর ভুল ধারণা থাকে যে বায়োলজি গেলে বোধহয় অঙ্ক করতে হয় না। তো তুমি ওখানকার কেমন দেখছো ব্যাপারটা?
হ্যাঁ, এই ব্যাপারটা মানে আমার আমার নিজেরও মনে অনেক সময় হয়েছিল যে হয়তো বায়োলজিতে ম্যাথমেটিক্স করতে হবে না, কিন্তু আমার নিজেরও 11-12 ম্যাথ ছিল, আর ম্যাথমেটিক্সটা লাগবেই। মানে তুমি সায়েন্সে পার্সু করতে গেলে হায়ার এডুকেশন, তোমার ইভেনচুয়ালি তুমি যাই করো, এমএসসি করো, বিএসসি, তারপর তুমি পিএইচডিতে যাও, ম্যাথমেটিক্সটা ইন সেন্স কি একটা নর্মাল মলিকুলার কনসেন্ট্রেশন, লাইক আমরা যেটা করি সিওন ভিওন C2B2, এই যে ক্যালকুলেশন, সেটার জন্য তো ম্যাথ লাগবে। সো এমন না যে ম্যাথটা কোনো সময় চাই, বা সায়েন্স মানে কিন্তু একটা বায়োলজি বা আলাদা কেমিস্ট্রি বা ফিজিক্স বা ম্যাথ এরকম আলাদা না। ওপরে গিয়ে যত তুমি ওপরের দিকে উঠতে থাকবে, তুমি দেখতে থাকবে সমস্ত কিছু সে এক জায়গায় এসে তোমাকে বিভিন্নভাবে পার্ট হয়ে গেছে, রিসার্চের হোক বা পড়াশোনার। সো ম্যাথটাকে এভাবে না নিয়ে তুমি 11-12টা ম্যাথটা রাখো, তারপর ইভেনচুয়ালি বিএসসিতে গেলে তোমার যদি বিএসসি বায়োলজি থেকে করতে চাও তো কেমিস্ট্রিটা রাখো, কেমিস্ট্রি থাকলো, বোটানি, জুলজি বা অন্য সাবজেক্ট যেগুলো রিলেভেন্ট, মাইক্রোবায়োলজি হতে পারে, বায়োটেকনোলজি হতে পারে। তো কেমিস্ট্রিটা থাকলেও হয় কি, কেমিস্ট্রি এমন একটা সাবজেক্ট যেখানে অনেক সময় রেকারেন্টলি ম্যাথ আসতেই থাকে, ক্যালকুলেশন আসতেই থাকে। তো এইভাবে ভাবতে পারো, ম্যাথ মানে এমন না যে আমাকে ওই ক্যালকুলাস আর ইন্টিগ্রাল ওইসব করতে হবে, কিন্তু ইভেনচুয়ালি ম্যাথটাকে অন্যভাবে অ্যাজ এ ম্যাথ মানে কি নাম্বার, মানে নাম্বার যেখানেই থাকবে কাইন্ড অফ ম্যাথ। সো ওটা ওইভাবে আসতে থাকলে কনসেপ্ট ওইভাবে রাখো যে ম্যাথটা অ্যাজ এ কেমিস্ট্রি ইজ অলসো ম্যাথ। সো ওইভাবে করলে জিনিসটা অনেকটা ইজি হবে ইন ফিউচার।
মানে আমরা ছোটবেলায় যেটা দেখেছি 90-এর দশকে ইন্ডিয়ান গভর্নমেন্ট খুব এমফাসাইজ করেছিল দুটো সাবজেক্টের প্রতি, একটা হচ্ছে কম্পিউটার সায়েন্স সিএস, আর একটা হচ্ছে বায়োটেকনোলজি, এবং বলা হয়েছিল 20 সাল নাগাদ বায়োটেকনোলজি এবং কম্পিউটার সায়েন্সের বিশাল ডেভেলপমেন্ট হবে। কম্পিউটার সায়েন্সের ডেভেলপমেন্টটা হয়ে গেছে, কিন্তু বায়োটেক ইন্ডাস্ট্রিয়ালাইজেশন কিন্তু
বায়োটেকও হয়েছে ব্যাপারটা, হ্যাঁ মানে আমাদের রিসেন্টলি আমরা কি
পৌঁছায়নি, কম্পিউটার
হ্যাঁ
সায়েন্সের মধ্যে পৌঁছায়নি।
রাইট রাইট। সো যেটা হয়, অনেকে মানে এগুলোকে বলে গ্ল্যামারাস জব, লাইক কম্পিউটার সায়েন্স, ডেটা সায়েন্স, এগুলোর মধ্যে একটা মানে গ্ল্যামার আছে, অনেক ব্যাপার আছে। কিন্তু বায়োটেকটা আমরা রিসেন্ট কি দেখলাম, যখন কোভিড আসলো, পুরো দুনিয়ার মধ্যে একটা হলো যে অ্যান্টিবডি চাই, অ্যান্টিবডি চাই। বাট চাই তো ঠিক আছে, বাট সেটার জন্য যে ফান্ডিং বা ওভারঅল রিসার্চ বা ম্যান ফোর্স, এগুলো তো আসবে এইভাবেই, মানে আমাদের আমাদের থ্রু দিয়েই আসবে। আমরা বেসিক বিএসসি করব, তারপর এমএসসি করব, তারপর পিএইচডি করব। সো আমাদেরকেই সেই রোলটা প্লে করতে হবে। তো সেটা মাথায় রেখে আমাদের নিজেদেরকে প্রিপেয়ার করা উচিত, যতটা নিজেদেরকে এনরিচ করা যায় বিভিন্নভাবে, বিভিন্ন মেথড শিখে হোক, বিভিন্ন ট্রেনিং নিয়ে হোক, বিভিন্ন কোচিং নিয়ে হোক, করতেই হয়। ইভেন আমি এখনো করে থাকি। তো এইভাবেই নিজেকে যতটা হয় প্রিপেয়ার রাখা উচিত।
এবং আজকের দিনে দেখছি বায়োলজিতে না ধীরে ধীরে হচ্ছে কম্পিটিশনাল বায়োলজি চাকরি বা অন্যান্য স্কোপ ধীরে ধীরে বাড়ছে। আমাদের
রাইট।
একটি স্টুডেন্ট রিসেন্টলি ইটালিতে মানে পরশুদিন চান্স পেলো, ও পেয়েছে ডাটা সায়েন্স ইন মেডিসিন, মানে
হুম হুম হুম।
বায়োলজিতে লাইফ সায়েন্স, কিন্তু ওর পিএইচডি অপশন হয়েছে ডাটা সায়েন্স ইন মেডিসিন
Right.
হিউম্যানিটিস ইউনিভার্সিটিতে।
হুম
তো
হুম।
ধীরে ধীরে বায়োলজির মধ্যেও কিন্তু কম্পিটিশনাল ঢুকে এই
একদম
সেক্টর
একদম
অপরচুনিটি বাড়বে, আমার যেটা ব্যক্তিগতভাবে মনে হয়। তোমার কি
একদম
মনে হয়?
একদম। হুম। একদমই। আমরা দেখো, আমরা মানে ইন্ডিয়ান, মানে আমিও ওইরকম একটা জায়গা থেকে এসেছি। আমি মুর্শিদাবাদে আমার বাড়ি ছিল, তারপর আমি ওয়েস্ট বেঙ্গলে সবকিছু জানি, ইভেন আমি স্টেট ইউনিভার্সিটি থেকেই পড়া। তো আমি যেটা মনে করি যে অ্যাজ এ বিএসসি বা এমএসসি লেভেলে তোমার কাছে যে রিসোর্সটা আছে, সেটা তুমি ফুল ওটাকে ইউজ করো। যা যা তুমি যতটুকু করতে পারবে 100% দিয়ে, যতটুকু জানা যায় সেগুলো করো। এবং তারপর হবে কি, তোমার কাছে অনেক অপশন থাকবে। যেমন দাদা বলছে যে বিভিন্ন এই যে কম্পিটিশন, ইভেন মানে সমস্ত বড় কিছু যে রিসার্চ গুলো, সেগুলো অনেক বায়োফিজিসিস্ট বা বায়োকেমিস্ট, তারা তারা এগুলো এসে আমাদের ফিল্ডে গিয়ে অনেকটা অ্যাডভান্স করে দিয়েছে। সো আমরা নিজেরাও সেগুলোকে ইজিলি অ্যাডাপ্ট করতে পারি। এমন কিছু আহামরি জিনিস না, কিন্তু নিজেকে সেই স্টেপগুলোর জন্য মানে বিএসসি থেকেই বা এমএসসি থেকেই নিজেকে একটু ইমপ্রুভ করতে হবে। মানে যত কিছু ভালো জিনিস নেওয়া যায়, যত ভালো কিছু শেখা যায়, সেগুলো শিখে, সেটা কোচিং এর থ্রু হতে পারে, কোথাও কোথাও ট্রেনিং নেওয়া হতে পারে, কোথাও কিছুদিনের জন্য ওয়ার্কশপ করা হতে পারে। এগুলো করে নিজেকে যতটা পারা যায় এনরিচ করো, যাতে ইন ফিউচার তুমি নিজেকে মানে অ্যাজ এ সামওয়ান ইউ ক্যান সে দ্যাট আই হ্যাভ দ্যাট এবিলিটি, আই ক্যান ডু দোজ থিংস। সো এটাই ম্যাটার করে।
আচ্ছা, কারো কিছু প্রশ্ন থাকলে, এই রিলেটেড কিছু প্রশ্ন থাকলে কেউ জিজ্ঞাসা করতে পারো। যদি কোনো প্রশ্ন মানে এই ধরনের প্রশ্ন থাকে সায়েন্টিফিক ছাড়াও,
একদম।
যদি এই প্রশ্নটাকে জিজ্ঞাসা করতে পারো। একজন আমি দেখলাম জিজ্ঞাসা করেছে একজন, আমি কোশ্চেন সেটের মধ্যে দেখছিলাম। একটা কোশ্চেন আমি তুলছি, ক্যান্সার রিসার্চের জন্য ভালো ইনস্টিটিউট কি ইউএসএ তে, প্লিজ বলুন স্যার।
এটা
এটা এটা তো ছোট
মানসিক, এটা
থাকে।
মনোজিৎ, এটা খুব খুব ভালো কোশ্চেন। বাট দেখো, মানে প্রত্যেকটা এখানে কি হয় বলতো, শুধু ইউএসএ না, আমাদের ইন্ডিয়াতেও অনেক ভালো ইনস্টিটিউট আছে এখন। বিশেষ করে তোমার ডিবিটি ইনস্টিটিউট গুলো, যেমন আমি নিজের অভিজ্ঞতা থেকে বলতে পারি, আমি যেখান থেকে পিএইচডি করেছি, দ্যাট ইজ এ ভেরি গুড ইনস্টিটিউট ফর ক্যান্সার রিসার্চ, অ্যাজ ওয়েল অ্যাজ আইএলএস ভুবনেশ্বর আছে, সিডিএফডি আছে, তারপর যেগুলো সিসিএমবি আছে,
আইজার
তারপর অ্যাক্ট্রেক আছে মুম্বাই ক্যান্সার রিসার্চের জন্য, খুব ভালো। টাটা মেডিকেল হসপিটালের সাথে কোলাবরেট করে করে কাজ ওরা। আইআইসিবি আছে কলকাতায়, যেখানে কিছু ল্যাবে ক্যান্সার রিসার্চ, খুব ভালো রিসার্চ হয়। তো এগুলো বিভিন্ন আছে। এবার তোমার যদি বাইরে আসতে হয় ইউএস এর জন্য, সেটা তোমার একটা লং টার্ম প্রসেস। সেখানে তোমাকে ওই এক্সাম গুলো যেগুলো দিতে হবে, আমি ওই প্রসেসের থ্রু আসিনি, বাট আমার যেটুকু নলেজ আছে, ওই টোয়েফেল বা এক্সাম গুলো দিতে হবে। তার সাথে সাথে তোমাকে, হ্যাঁ, ল্যাব স্পেসিফিক চুজ করে তোমাকে কভার লেটার লিখতে হবে, প্লাস সিভি বানাতে হবে সেইভাবে, যতটা প্রিপেয়ার করা যায়। তো এই জিনিসগুলো আছে। কিন্তু অবভিয়াসলি বাইরেও তুমি দেখতে পারো, যেখানে ক্যান্সার, এখানে মোটামুটি সমস্ত ইউনিভার্সিটিতেই, এমন না নামের ওপরে যেও না, ল্যাবের বেসিক্যালি ল্যাব, যে ল্যাবটা তুমি অ্যাপ্লাই বা ইন্টারেস্টেড যে টপিকের ওপর, সেই ল্যাবটা কোথায় ভালো আছে, কোন গাইড বা কোন সায়েন্টিস্ট এটার উপর ভালো রিসার্চ করে, সেটা একটু দেখো। সেভাবে তোমাকে করতে হবে, অ্যাপ্লিকেশন গুলো করতে হবে।
একদম একদম। না আমরা মানে তোমার এতটা সময় পেয়ে মানে আমরা সত্যি খুবই আপ্লুত এবং কৃতার্থ। মানে আমাদের বাচ্চা যারা
না,
দেখছেন,
আমারও
তাদের মধ্যে
ভালো লাগলো পুরো, হ্যাঁ, শেয়ার করে সমস্ত।
হ্যাঁ,
তাই
মানে
না?
তারা তারা কলেজে বা ইউনিভার্সিটি বা স্কুলে পড়ে, বা অন্যান্য দর্শক যারা আছেন তারা
হুম হুম হুম।
দেখছেন, তাদের মানে কাছে এটা মানে কি বলবো, অসাধারণ পাওনা। এতক্ষণ ধরে মানে আমরা প্রথমে, সত্যি কথা বলতে প্রথমে প্ল্যান করেছিলাম একটা 1 ঘন্টা সেশন করবো, সেটা হতে হতে 2 ঘন্টা সেশনে পৌঁছে গেছে। হ্যাঁ, মানে
হুম,
সেটা আমাদের কাছে অসম্ভব ভালো পাওনা। তো অনেক 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 ধন্যবাদ মানে এই এই এতটা সময় দেওয়ার জন্য এবং প্রত্যেকটা টপিক এত সুন্দর করে বুঝিয়ে বলার জন্য। হ্যাঁ,
থ্যাংক ইউ,
তো
থ্যাংক ইউ।
আমরা আশা করবো অদূর ভবিষ্যতে আরো একদিন আমরা এরকম ভাবে বসবো
একদম,
একসাথে
অবশ্যই, অবশ্যই,
এবং
অবশ্যই।
সবাইকে নিয়ে এরকম ভাবে বসবো এবং এর মাধ্যমে আমাদের সত্যি কথা বলতে বাংলা বা ওয়েস্ট বেঙ্গল বা আমাদের দেশের বিজ্ঞান সমৃদ্ধ হোক এবং আগামী দিনে আমাদের দেশের বা আমাদের রাজ্যের সমস্ত যারাই হোক মানে দেশ রাজ্যে তো কোনো বাউন্ডারি নেই মানে পৃথিবীর
না,
বিজ্ঞান উন্নত হোক, পৃথিবীর সবাই মানুষ বিজ্ঞান চর্চায় করুক ভালোভাবে করুক এবং ভালো কাজ নিয়ে আসুক যেটা দেশের দশের কাজে লাগে একদম।
একদম কারেক্ট।
সেটাই হবে আমাদের এই এই যে সম্প্রসারণ গুলো হচ্ছে বা এই যে পডকাস্ট মানে পডকাস্ট ইংরেজি শব্দ তার বাংলা ছিল সম্প্রসারণ। হ্যাঁ, তো এই যে সম্প্রসারণ গুলো হচ্ছে সেটার মূল উদ্দেশ্য এটাই মানুষকে ভালো বিজ্ঞান চর্চায় মানুষকে অনুপ্রেরণা দেওয়া এবং সেই অনুপ্রেরণা দেওয়ার জন্য সুভাষিসকে অসংখ্য অসংখ্য ধন্যবাদ মানে আমাদের
ধন্যবাদ তোমাকেও। হ্যাঁ,
হ্যাঁ, তো আজকের মতো আমরা এখানেই আস্তে আস্তে শেষ করব সবাইকে শুভরাত্রি কিন্তু সুভাষিস শুভরাত্রি নয় সুভাষিস তোমাকে বলতে হবে হ্যাভ এ গুড ডে। হ্যাঁ,
থ্যাংক ইউ, থ্যাংক ইউ।
সেখান থেকে বেরিয়ে সোজা ল্যাবে দৌড়াবে আমি জানি। হ্যাঁ, শুভাশিস তোমার শেষ কোনো কথা ওনাদের উদ্দেশ্যে সবার উদ্দেশ্যে।
থ্যাংক হ্যাঁ, সবাই বাস হ্যাঁ, সামনে পুজো আসছে, ভালো করে কাটাও। আর হ্যাঁ, অ্যাজ এ এই ক্যারিয়ারটাকে অ্যাজ এ লং রান ভাবো, কোনো কিছুই মানে খুব তাড়াতাড়ি হয় না, একটু টাইম নেয়, মানে ধৈর্য নেয়, সবকিছুতে ধৈর্য রাখতে হবে। আর যতটা পারা যায় নিজের মতো করে চেষ্টা করে যাও, বাকিটা তো হয়েই যাবে ইভেনচুয়ালি। কিছু না কিছু ভালো হবেই, ভালো চেষ্টা করলে তার ফল ভালো হবে।
একদম
বাস
একদম।
এটাই। ঠিক
আজকের
আছে,
মতো আমরা বিদায় নিচ্ছি সবাইকে।
ধন্যবাদ।
ধন্যবাদ
থ্যাংক ইউ,
ধন্যবাদ
থ্যাংক ইউ।
সুভাষিস আবার
শুভরাত্রি।
শুভরাত্রি।